Have you ever wondered how the pigeon got its head crest? Okay, so maybe not, but I'm sure you have thought about other birds and their head crests. But the rock pigeon has a diverse range of head crest styles, and I'm sure you'd like to know more. The geographic origins of breeds and the genetic basis of variation within the widely distributed and phenotypically diverse domestic rock pigeon, or columbolivia, remain largely unknown. But a recent study done by Michael D. Shapiro, Genomic, and a team of scientists from around the world may have shed some light on the matter. To determine the relationship between species, they examined genomic diversity, genetic structure, and phylogenetic relationships among domestic breeds and feral populations, being free-living birds descended from escaped domestics. From this, a phylogenetic tree was created to display the found relationships, as you can see here. To discover the probable ancestry and resultant genomic diversity, they examined the genetic structure among breeds by analysing 3,950 loci with admixture, which is a software tool for maximum likelihood estimation of individual ancestries for multi-locus single nucleotide polymorphism (SMP) genotype datasets. From the resulting data, Shapiro and his team found a best model fit at k equals 1, where k is the number of assumed ancestral populations. Now that they had discovered the genomic diversity of the rock pigeon, Shapiro and his team turned their focus to the evolution of the head crest. They resequenced 8 crested individuals to test whether the crest development is controlled by the same mutation in separate breeds. Then sorted genomic variants from birds with and without head crest into separate bins and calculated allele frequency differentiation, or FST, across the genome, identifying a region of high differentiation between crested and uncrested birds in the pigeon ortholog of ephrin receptor B2, or EPHB2. The role of EPHB2 in feather growth is unknown, but it plays an important role in tissue patterning and morphogenesis, and is a member of the receptor tyrosine kinase family that mediates development of the feather cytoskeleton. All 8 crested birds were homozygous for a T nucleotide at scaffold 612, position 596613, hereafter the CR allele, whereas uncrested birds were heterozygous or homozygous for the putatively ancestral C nucleotide, or the plus allele. Shapiro and his team then used the Variant Annotation Analysis and Search tool, or VAST, to investigate the pigeon genomes for additional coding changes associated with the head crest phenotype. This identified one gene with genome-wide significance, EPHB2, and specifically the CR single nucleotide polymorphism, or SMP. Although the results were not conclusive, Shapiro and his team concluded that pigeons were a model for identifying the genetic basis of variation in traits of general interest. It was also concluded that pigeons are a promising model to explore the genetic architecture of derived constructive phenotypes in a bird that is open to genetic, genomic, and developmental investigation.